Hello everyone, welcome to today's live broadcast, Simplified Imaging for Quantifying Biological Complexity of iPSC-derived Cardiomyocytes and Neurons. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, the leading social media site for science professionals, and sponsored by Molecular Devices. Molecular Devices is one of the world's leading providers of high-performance bioanalytical measurement systems, instruments, software, and reagents for life science research, pharmaceutical, and biotherapeutic development. Before we start, there are a few instructions. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenters. We are proud to welcome Grisha Chandy, PhD, Senior Product Manager at Molecular Devices, and Oksana Sirenko, PhD, a Research Scientist at Molecular Devices. Grisha's scientific background is in cell biology, physiology, and biophysics. He has used and developed imaging techniques and technologies throughout his career, starting with his doctoral work at UC Irvine, his postdoc at UC Berkeley, as well as research scientist position at Stanford University. Grisha has been with Molecular Devices for 10 years, driving the launches of 4 ImageXpress microsystems. They include the ImageXpress Microconfocal System, MetaXpress Software versions 3 onwards, and most recently, the launch of the ImageXpress Nano system and CellReporterXpress Software. Oksana is working on development of new technologies for imaging and high-content analysis. She gained experience working on drug discovery and drug development at several biotech companies in the San Francisco Bay Area. The focus of Oksana's current scientific research is on development methods for high-throughput in vitro toxicity screening, including 3D models. Oksana has a PhD in biochemistry. I will now turn it over to Grisha Chandy for his introduction.
The agenda is that I, Grisha Chandy, Product and Applications Manager for Cell Analysis at Molecular Devices, will host and be the Q&A moderator. I will also introduce you to Molecular Devices automated imaging systems. I will then hand over the webinar to Oksana Sirenko, Senior Research Scientist here at Molecular Devices, who will walk you through our new software, CellReporterXpress, with a workflow focused on routine imaging assays, and she will demonstrate and show some data on multi-parametric assays for compound screening and toxicity assessment with those same iPSC-derived cells I just mentioned. So a brief introduction to Molecular Devices automated imaging systems. So the idea is to take what you would do with a conventional microscope, which is have a sample. It might be a slide, it might be a multi-well plate, put on a microscope, take an image, try to make sense of what that image is saying in an unbiased method, and make sense of the measurements you're making. And unfortunately, with a manual workflow, the elapsed time is typically hours. Now, the idea with Molecular Devices products, the ImageXpress systems, is to scale that up to be able to use multiple slides, multiple plates, put them in your system, do the variety of assays, and in an unbiased way make those measurements and make sense of that data and drive that manual workflow from hours to minutes. Molecular Devices has a variety of ImageXpress Systems to handle a variety of workflows that you might be experiencing, all the way from the ImageXpress Micro Confocal that is optimized for 3D assays, the ImageXpress Micro 4, which is a robust system for very high throughput assays or live cell assays, and the new ImageXpress Nano system that uses new software that's called CellReporterXpress, which Oksana will be introducing you to. And here I want to highlight the ImageXpress Micro live cell assays that are potential to do with our systems. This apoptotic inducer assay in a multi-well plate where you're watching these cells over time, and you're not staying in one well, you're going to multiple wells over and over again. So we have apoptosis, you have some cell proliferation, you have bacterial phagocytosis, you have cells migrating with a variety of sensors in them, you have a multicolor assay going on over time lapse. So these are all possible with the ImageXpress Micro systems. So at this point I want to introduce the ImageXpress Nano system, a cost-effective automated imager for every lab, ideal for routine assays. It's available with 2x to 60x objectives, 5 filters, solid-state light engine with a very long expected lifetime, and it comes with 2 options available: transmitted light and or environmental control. The product has a large field of view, just like our other ImageXpress systems, better than 25 nanometer X, Y, and Z stages, high image quality, the new easy-to-learn software, automatic stitching and montaging, on-the-fly image analysis, and label-free transmitted light analysis. So the CellReporterXpress software is easy to use, a browser-based interface, tablet-friendly, touchscreen compatible, no database, and access and share your data anywhere at any time. You can use to acquire data, analyze remotely, share, and then present your data to your colleagues. Note, I'd like to turn this presentation over to Oksana Sirenko, who's going to tell you more about the applications that are possible to do with this, the new ImageXpress Nano system using CellReporterXpress software.
Thank you, Grisha, for your introduction. Let me start with a short introduction for CellReporter software. In the home page, you can see the icons for acquisition, analysis, data review, monitoring, or configuration. Let's go to the acquisition. You may start from a protocol template. First, you select the instrument, then go to the settings. You may select plate types, different colors, or magnification. Then you can acquire the image. Instrument will take images and present it as a composite image. You can modify the exposure time or focus offset for different stains. You can select or modify the area of your interest. You can set up the analysis on the fly. To set up the analysis, go to the list of analysis templates and select the one, for example, cell scoring. The analysis counts and analyzes cells using intensities in different colors. Then you can select the area for analysis. Finally, you can select the wells to acquire. You can save your protocol and create the data file. You may check the settings before acquisition and then hit the Run button. Image analysis allows characterization of cells and various markers. You may start from preselected analysis template or create your own. There are more than 20 predefined analysis protocols, including cell viability, differentiation, mitochondria, cell cycle, and many other applications. Here you can see the images of cells stained with 3 dyes. nuclear dye in blue, phalloidin in green for cytoskeleton, and mitotracker orange dye for mitochondria. For the analysis, you can select from a variety of available protocols. For example, select cell scoring analysis. Cell scoring analysis is based on finding nuclei, then scoring cells as positive or negative for the appropriate marker. Nuclei or cells defined by dimensions, minimum and maximum, also by intensity threshold over the background. You may start from the preselected numbers, or you can modify those by typing desired values. You may use automatic or manual definition. For example, in the auto mode, you can just click to find the objects, nuclei. You can use manual option to trace desired objects, for example, cells. Finally, you can run test analysis and see the masks of the objects and also the measurements. After analysis is done, you can select graphical presentation, dot plots, heat maps, bars, or tables. The next part of our presentation is dedicated to multi-parametric assays for compound screening and toxicity assessment. The latest trends in biology highlighted by increase in complexity of assays that includes complex cell models and phenotypic multiparametric readouts. The instruments have to enable increasing complexity of assays and also ease of use and throughput. The flowchart for the typical automated imaging assay includes cell plating, treatment them with compound, staining, then imaging. Images analyzed by software and allowed to derive multiple measurements that would characterize the effect of compounds on cell morphology. For example, for multi-parameter cytotoxicity assay, calcium AM can be used for defining the number of live cells, also cell morphology characterization. Nuclear dye can be used to determine nuclear intensity, shape, or apoptosis, and mitotracker orange used to measure mitochondria potential or autophagy. Cells can be imaged live after one single step of dye addition. Also, you can use fixed cells protocol to evaluate additional markers, in this case, cytoskeleton. Cell analysis quantifies biological effects for multiple measurements. Multiparametric assays are especially useful for description of complex effects, subtle changes, and allow to describe various phenotypic effects. For example, you can observe very subtle effects of compounds, in this case, to spot apoptotic cell. By measuring cell area or signal intensity, you can describe changes in cell spreading and also changes in cytoskeleton, in this case with paclitac cell. You can spot changes in a number of nuclei or nuclear shape. You can also measure cell shrinking or cell detachment, in this case with staurosporin. Finally, you can use multiple measurements to define AC50s for different compound effects. In collaboration with Cellular Dynamics Company, we developed toxicity assays for stem cell-derived cell models. Cardiotoxicity assays are extremely important for drug safety evaluation, and neuronal toxicity assays can be used for a variety of applications, including disease modeling or testing chemicals for the possible impact on neurodevelopment. We described in the past the functional cardiac assay using calcium fluxes. This assay can be done on several molecular devices instruments, including flipper, plate readers, and also imaging instruments. Cell morphology evaluation brings another level of important information. We developed methods for either live cell or fixed cell toxicity evaluation. Cell staining includes the combination of several dyes, as we described before. Here you can see the images of control cardiomyocytes and cells treated with staurosporine or doxorubicin. We tested the panel of known cardiotoxic compounds, and to evaluate cardiac toxicity, cells were treated for 24 hours. Readouts include nuclei count and intensity, also cell area and stain intensity, and also the numbers and percentage of positive or negative cells. For cardiotoxicity assay, live cell analysis or cell scoring analysis was used. In a drop-down menu, you can select or add different measurements. You may select particular wells for review of images. You may see control wells, also wells treated with cardiotoxic compounds like amiodarone, doxorubicin, staurosporine. Also, you may review the analysis masks for control and other compounds. We defined EC50s for cardiotoxic effects using the number of positive cells and the area of positive cells. The assay allowed detect cardiotoxicity and also rank compounds for their effect. We have used stem cell-derived neurons for neurotoxicity assays. There is an increasing concern about the large number of untested chemicals in the environment, and there is a pressing need to develop reliable and efficient screening tools to identify chemicals that could affect human health. We evaluated neurite outgrowth assay as possible screen to explore ability of compounds to adversely affect the developing nervous system. Neurons were treated for 70 hours with various compounds, and effects on neurite outgrowth were evaluated. This is the picture of eye cell neurons in a culture stained with calcium AM. Neurite tracing analysis allows to define cell bodies, neurite outgrowth, numbers of processes, and branching points. Here you can see the screenshot of the entire plate for neurotoxicity assay. Neurite tracing protocol was used for image analysis. In the data review mode, you may see the images or numbers. Different readouts can be selected for the heat map, neurite outgrowth, number of branches, or others. You can see those responses for compound effects. Then, if you go back to the images, you may review untreated samples and samples treated with compounds, for example, methyl mercury. Analysis masks can be visualized for outgrowths, cell bodies, and branches. Then you may go to the graph presentation, for example, neurite outgrowth versus branches. You may highlight those dots and then go back and forth between the graphs, images, heat maps, and the table. Here you can see the images and tracings of neurites treated with compounds known for their neurotoxicity. Dose-dependent disintegration of networks observed. We have created IC50 plots for multiple readouts. The IC50s for multiple measurements presented in the table. Notably, the IC50s were different for different readouts. For example, IC50 for branching were typically the lowest, and for cell viability significantly higher than the outgrowth, branching, or processes. So, stem cell-derived assay models in combination with advanced imaging methods can be a very efficient tool for toxicity evaluation. Finally, I would like to describe several other assays that can be used for a variety of applications. One is cell count and characterization in transmitted light. The analysis can segment and accurately count cells in transmitted light that would allow to monitor cell proliferation, toxicity, or migration label-free without using any dyes. We demonstrated the accuracy of analysis by comparison with nuclei count using fluorescence. As you can see, numbers were very close for different cell concentrations. Also, we were able to measure toxicity effects measuring cell numbers and area in transmitted light. You can also combine transmitted light with fluorescence and run cell scoring analysis. That can be used for evaluation of transfection efficiency and many other applications. DNA damage is another important application for toxicity assessment. Assessing damage to DNA is frequently addressed with research applications because of its implications in diverse diseases, including genetic mutations, cancer, and aging. DNA damage may happen spontaneously or be a result from exposure to ionizing radiation or chemical compounds. Cellular DNA damage can be visualized and quantified using antibodies against phosphorylated H2AX protein. Also, we have used nuclear stain HERST for counterstaining. On-the-fly cell scoring analysis efficiently identifies nuclei with damaged DNA. Then, CellReporterXpress Software includes a preconfigured cell scoring application model. That model can classify nuclei for positive or negative for the marker. Imaging was done with 10x objective, and it was sufficient to calculate accurate concentration responses curves. You can see those responses for mitomycin or etoposide. Autophagy is a regulated process of degrading and recycling damaged proteins and organelles in response to cellular stress. This regulation of autophagy has been established to play a role in various neurodegenerative diseases and also cancers. For the assay development, we were using PC12 neuroblastoma cell line. And it is very important to define those small objects by imaging. You can see here autophagy particles stained with cyto-ID, and also mitochondria particles stained with mitotracker orange. We measured the number, area, and intensity of autophagy particles and compared effects for several inducers of autophagy. And finally, count and characterization of small particles allows to measure GPCR activation by agonist or antagonist in the Transfluor assay. Transfluor assay uses cell lines which overexpress GPCR gene of interest and also fluorescently tagged beta-arrestin. The predefined pits and vesicles analysis model can identify and count pits and vesicles which form by beta-arrestin translocation. We presented here a new imaging system and software that enable imaging and analysis of complex biological responses in automatic screening format. Those complex multi-parametric measurements allow better characterization of biology and compound effects. Thank you so much for your attention.
I'd like to thank you for your time. If you have additional questions for the speakers, our emails are given right there, grisha.chandy@moldev.com and Oksana Sirenko @moldev.com. And if you want to learn more about our products, please visit us at www.moleculardevices.com, Systems High Content Imaging. And now we would like to open it up to questions from the audience. Please let us know if there is anything that you would like more information about.
That information to us. So before we start with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. And before we begin the question and answer session, I want to advise that a series of poll questions will appear on your screen during the Q&A. You may select your answers, then close the poll questions by clicking on the X in the right corner. Grisha will handle the Q&A, and I will hand it off to you, Grisha.
Thank you very much, Judy, and thanks, Oksana. I'll just give you a few seconds to answer those poll questions that might be popping up, and then I'll just sort through these the questions that have come in during the webinar. And if you have additional questions, please submit them, and we will, as she said, we'll try to get to them. Oksana, what magnification is better to use for a neurotoxicity versus a cardiotoxicity assay?
So in majority of my assays, I use 10x, standard 10x magnification, which allows to enable high throughput, because with this magnification you need to capture just kind of one image per well in order to have a sufficient number of cells. But if you're interested more in kind of subcellular structures, I would recommend using 20x magnification for neuronal cells, and in some cases for cardiac cells as well.
There are a couple of questions about transmitted light. And I'll ask both of them. One question is how well it works with different cell types, and the other one is whether you can explain a little bit about how the software identifies cells accurately using the transmitted light.
So the first question is about whether it works good with different cell types. In fact, the software has slightly different application models which are applicable for different cell types. So there is one application model which is general use, and that one kind of works good with majority of common cell types. Like we evaluated it with the HeLa cells, with CHO cells, with cardiomyocytes. We get pretty accurate cell count and also area count evaluation of covered area. So sometimes cells could be smaller, and for example, these blood cells, or bigger, like PC12 cells, or some other cell types like hepatocytes. For those cell lines, it's better to use slightly different application models, which you kind of call like large cells or bead count. Those can be also used for rounded up non-adherent cells or for other cell types. So basically we recommend that different, so you start from general application model, and if not satisfied with results, you can use different one and pick which one works better for your specific conditions. It is very important also for transmitted light count to have focus, right? So that cells appear crisp and kind of very nice contrast. So that could be adjusted by modification, some acquisition parameter like focusing or exposure time. So, and also the second question was about algorithm which segment. I believe the algorithm use the features of cells, such as their brightness or contrast or their size. And we have algorithms which based on kind of predefined settings, which kind of works for specific cell types or specific cell measurements. And some other algorithms, they include machine learning components which could be adjusted for your specific cell types.
Great. Thank you, Oksana. There's a couple of questions about the type of samples we can put into the instrument. One asks about whether you can do slides, and the other asks whether you can do tissues. And can you answer those questions?
Mm-hmm. So the analysis, both acquisition and analysis, are suitable for the slide format. So it has a settings for slides where you can capture entire slide, and actually the features of software where you can it can be very flexible with the size or kind of area you acquire. So you actually don't have a limitation which object you want to capture, so which kind of works very well for slides. So first you might capture with a greater magnification and then zoom in and capture your area of interest on a slide. And for analysis, you can use the same modules as for regular cells, and it would be kind of no difference in handling this type of analysis. So different sites which would be stitched together and analysis done as such that you wouldn't see even seam between different sites when you acquire those samples.
At what day were the neurotoxic compounds added for the iCell neuron experiments?
Yes, this is actually a good question. So in my we published this type of assay previously, where we used 48 hours period between plating of neurites and then addition of compounds. In this recent experiment, I added compounds 24 hours later, and then the culture was going to 3 days, and it's also possible to continue a culture for more time, like for 5 days or 7 days, to evaluate not only neurotoxicity but also neurodevelopment component in this assay.
So I'm just reading through the questions. How do you analyze the mitochondrial integrity, and what magnification did you use for that?
Yes, so for mitochondria evaluation, actually 2 different approaches could be used. So for really high throughput mode, where you would like to use like 10x magnification, I would recommend to use cell scoring analysis, so which works to identify intensity and area and score cells for positive or negative in terms of intact mitochondria potential. And there is also another approach could be used, which would deliver more information about and more sensitivity for mitochondria assay. In this approach, I would recommend to use 20x or 40x magnification and actually count mitochondria granules, and not only count those using good granularity application model, but also to define their intensities and area for those granules. So that model works very well for mitochondria toxicity and also autophagy applications. So, but you just have to keep in mind that if you use higher magnification, it is important to acquire more than 1 site per well. So usually we use like 2 or 4 sites if you use 20x magnification, or we typically use 4 sites if we use 40x magnification. That way you will get sufficient statistical to cover cells and for variability in cells to cells.
There are, thanks Oksana again. There are 3 or 4 questions about the calcium assay slide you threw up. Mm-hmm. I'll answer one of them, and then maybe you can talk a little bit about the assay. So the questions are, what is the acquisition rate for dynamic cardiac? What's the acquisition rate for that assay? How are the calcium imaging data for neurons and cardiomyocytes analyzed? And is this something like what you do for the flex station? So the data that Oksana showed for the calcium was acquired with our ImageXpress microsystem. Most of the data, all the data in this presentation besides that, was from our new ImageXpress Nano. So on the ImageXpress microsystems, you can acquire data at a variety of frame rates all the way up to 100 frames a second, or even faster, potentially possible if you use a smaller area of the acquisition window. But maybe you can talk a little bit about, I think you published some paper, and how you do the actual assay.
Mm-hmm. So actually, we published several papers already on cardiac toxicity using calcium flux method. Most of this work was done on a flipper, so we have like 2 papers in 2013 and some recent papers as well in 2016. And kind of recently, we published a paper on cardiac spheroids in Assay Drug Development Technology. And in that paper, we actually describe in detail how you can do this assay using imaging instruments. So right now, we have the algorithms which can analyze the calcium fluxes the same way as it's done on a flipper instrument. So you can define amplitude, beating rate, also distance between peaks, peak width, and other parameters. So, and there are kind of protocols which describe in that paper from 2017, which you can use, or you can contact our technical support for more information how to use this method. So what's important about speed of acquisition? So for calcium flux assay, especially if you use at least cells from Cellular Dynamics International, we typically use like 8 frames per minute, 8 frames per second, I'm sorry. And also you can even go slightly lower than that, like 3 frames per second. In an ImageXpress Micro, I typically use like 10 frames per second for this calcium flux assay.
But the systems are capable of a higher frame rate acquisitions.
Yes, you can do up to higher rates. Yeah.
If there's any other calcium questions, please go ahead and send them in. A question came about that I'll take about the CellReporterXpress and what systems it's available on. Yes, the CellReporterXpress, our new software, is available only on the ImageXpress Nano at this time, and our ImageXpress microsystems are continue to use our MetaXpress software. And I don't see any additional questions coming in, and so I'll give it a couple of minutes. Otherwise, I'll turn it over to back to Judy to close out the webinar.
You know what, Grisha, if you would like, we can allow you to say a few things, and while you are, if questions come in, then when you're done, you know, you could jump onto those questions. Does that sound good?
That sounds fine. Oksana would like to say something.
Yes, so I would like to use this opportunity to acknowledge again our past and present collaborators, especially Cellular Dynamics International, and also our collaborators from Institute of Environmental Health.
So thank you. I just want to thank people for coming and attending our Medical Devices webinar, and I hope to see you again at a future webinar of ours.
And I would like to once again thank Grisha Chandy and Oksana Sirenko for their presentation. I would also like to thank our sponsor, Molecular Devices, for making it possible to bring this presentation to you. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Today's webcast will be available for on-demand viewing through May 2018. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. We will see you next time.